welcome everybody to the combined 2020-2021 Charles DeLisi Distinguished Lecture. As you all recall, Ioannis Paskalidis was identified last year. We were all set to go in April, and then a pandemic hit the world, and we all had to go remote, and we held off on virtually everything scheduled at that time, and now we're starting to re-engage. We've now got fully over that trauma and have a good sense of how to interact on Zoom and have great lectures and events on Zoom as best as we can. So we're going to proceed, of course, and have Ioannis give his lecture today. This lecture is named after Charles DeLisi, who endowed the lecture series. Charles is currently the Dean Emeritus of the College of Engineering. He was the Dean for a decade. And absolutely transformed the College of Engineering to move from primarily an undergraduate engineering college to a major research college of engineering, changed the culture, changed the faculty we were able to recruit, and moved the whole college to the higher echelon of being a research one EAU level College of Engineering. And also in the process, was able to bring bioengineering down to the molecular and cellular level, and was the originator of the bioinformatics program here at Boston University. He is the winner of the President's Citizens Medal, awarded to him by President Clinton for his role in basically initiating the Human Genome Project, often called the father of the Human Genome Project. And we're very grateful to Charles for endowing this lecture series. In the past, to this point, just a quick announcement, a couple of quick announcements, but one point I want to make here, up to now, all of the Charles DeLisi lecturers have been faculty at the College of Engineering. We will start to do an alternation of faculty and distinguished alumni also that came out of our program and are now impacting their field using their foundation at Boston University. So Charles, thank you, and we will move on. Now, before I move on, or even before I do anything, there we go. A couple of reminders. If the faculty on this, remember there's a faculty meeting on Monday at 9 a.m. I'll go through this current state of the college and bring you information about university and college as we try to come out of COVID and prepare for the fall, including budget information and so forth. I'll remind everybody then again that there are several graduation events going on. We will have a virtual master's and PhD hooding event on Friday the 14th. And a formal graduation is on, for the whole university live is on that weekend. But we will have an undergraduate celebration block party on Thursday, May 13th at 10 a.m., which will have special awards for Doug and Kathy announced, as well as other awards for faculty and students, some fun and some videos created by both faculty and students. So please plan on attending that. Their parents will be there as well. Let's start out with the winners of the Early Career Award for Excellence in Research. We've had this award going on, I think, since 2008 or so. We had a winner last year. We have winners this year. In 2020, had we had the event, we would have announced Manuel Igli from Electrical Computer Engineering. Manuel has done a spectacular job during his early years as a faculty member with great success. His area is system software security, web security, privacy on mobile systems and online networks. He's an editorial member of the ACM Transactions on Security and Privacy. He's had several distinguished paper awards. This is just one of them. Over 55 publications in peer-reviewed places since he started here at BU, and many of his works getting highly cited, and he's impacted the field significantly. And he's a very well-funded faculty member of the College of Engineering. So congratulations to Manuel, and a virtual clapping to you. Well done. This year we had another great slate of people. It's getting harder and harder to identify. We've had some spectacular junior faculty in the last several years. And so we decided to give it to two faculty. The first one is Lei Tian from Electrical and Computer Engineering. He does work in computational imaging and sensing, particularly imaging around in a scattering media. He's had applications in both deep learning and in neurophotonics. Computational imaging is transforming many fields. Won career awards, best paper awards. He's been selected to be at the Frontiers of Engineering Symposiums, tremendous amount of publications for junior faculty, and many citations, again, highly impacting the field and substantial funding from both NSF and NIH. Congratulations, Leigh, to you. The winners of this award do get a $1,000 check. So congratulations to you, Leigh. I find clapping on Zoom to be really very sorry event. And then our second winner this year is Keith Brown from Mechanical Engineering and also Material Science and Engineering. Keith does work in hierarchical soft matter, autonomous experimentation, scanning, and probe techniques. He's won a Frontiers of Materials Award. He's been elected Professor of the Year in Mechanical Engineering. So he's a great researcher and a great educator. He's been a feature stars of the ABS. He's gotten many publications, 26 since beginning here at BU, and multiple citations, high H index impact in the field, and high success of funding from multiple agencies. So great success from these two wonderful junior faculty members, setting examples for all the junior faculty we've been hiring. So thank you and congratulations to Keith. Okay, so now we move on to the star of the show. Our 2020 and 2021, the only faculty to get this award 2 years in a row is Dr. Yanis Paskalidis of the Electrical and Computer Engineering Department of SAIS. So Yanis is a professor in electrical and computer engineering. He's a professor in systems engineering and biomedical engineering and computing and data sciences. He's also the director of the Center for Information and Systems Engineering. He obtained his diploma from the National Technical University of Athens in Greece, his MS and PhDs from MIT, all in electrical and computer science, electrical engineering and computer science. He's been at BU since 1996. His current research is in the field of systems and control, optimization, machine learning, networks, computational biology, and computational medicine, spearheading a lot of our efforts in digital medicine. He's the author of a monograph of over 220 refereed publications on these topics with over 130 invited lectures, and he's been the primary advisor of 26 of our PhD students. His work has been recognized early on with a Career Award from the National Science Foundation, the second prize in the 1997 George E. Nicholson Paper Competition by Informs. He's been the recipient of the IBM IEEE Smarter Planet Challenge Award, and a finalist Best Paper Award at the IEEE International Conference on Robotics and Automation. His work on protein docking with other collaborators has been recognized for best performance in modeling selected protein-protein complexes against 64 other predictive models. The 2019 Protein Interaction Evaluation Meeting selected this. His work on computational medicine won an IEEE Computer Society Crowdsourcing Prize and a Best Paper Award by the International Medical Informatics Association. He was an invited participant at the Frontiers of Engineering Symposium, organized by the National Academy of Engineering, and the 2014 National Academy's Keck Future Initiative Conference. He is a fellow of the IEEE and was founding editor-in-chief of the IEEE Transactions on Control of Networks and Systems. So I will introduce his talk, and if you have questions, please enter them in real time on the chat, or I'm sorry, the Q&A. Professor Clem Carl, the chair of ECE, will keep track of the questions and monitor the questions for Yanis after the talk. But Yanis today will be speaking about data science and optimization adventures in computational biology and medicine. I will unshare the slide. Yanis, the floor is yours.
All right, thank you, Ken. Let me share the slides. Okay, and just to make sure you see the slides and you see the laser pointer. Excellent, thank you. All right, so it's a great pleasure to be given the opportunity to give this lecture, and I would like to thank Ken and the college for selecting me. And it is no exaggeration to say that I'm really standing on the shoulders of giants, starting with Charles DeLisi, whose name graces this award, and also a number of colleagues that they are all research giants who have given this lecture before me. So in this lecture, we will talk quite a bit about predictive modeling. So I thought we should start with a warm-up exercise. So when I see lists of this type, I tend to think of these lists as data sets. So suppose that these are data points, different data points in a data set. And suppose that we are interested in predicting the mode in which they gave their lecture. So if we are to label this data set, so all of these were in-person lectures, and there are obviously implications about the state of the world at the time. And I have an interesting new data point in this data set, which is this one here. I don't even want to speculate what a predictive model would say, but it may potentially indicate that the college made a huge mistake. Nevertheless, let me remind you that association does not necessarily imply causation, so do not send me threatening emails. Moving on, and with that comment, so this is what comes next. So I'll take you through an adventure ride through a variety of systems, and the systems are ordered in terms of their scale, starting from molecules. So these are proteins, particularly a protein complex, going on to talk about cellular organisms. So this is a cell of a bacterium, and then to more complex organisms, larger organisms like humans. And in terms of methodologies that we are going to discuss in this lecture, so this is a snapshot of some of the methodologies that we will consider. And this adventure is going to evolve roughly like this. There are going to be 3 stops along the way. So let's start with the first stop, or the first item in this adventure ride. And we'll start with proteins. So proteins are large molecules. They are produced in the cell from a cellular machine, which is called the ribosome. And they are essentially a sequence of amino acids. There are 20 amino acids in nature. And as they get produced in the cell, they very quickly, this long sequence, folds and assumes a 3-dimensional structure. And it is the sequence of the protein that determines the structure, and it is the structure that typically determines the function of the protein within the cell. Now, proteins interact all the time. In fact, most cellular functions involve protein interactions. And when, for instance, we have 2 proteins that come together to bind and form a complex, typically the larger protein, we call it the receptor, and then the smaller protein, we call it the ligand, and they come and bind to form a protein complex. And the docking problem that will be of interest to us is the problem of predicting computationally what is this structure of the complex, assuming that we know the 3-dimensional structure of the component molecules that form this complex. So this problem can actually be thought as an optimization problem, and a hard optimization problem at that. From very basic thermodynamics, the proteins bind in a way that minimize the so-called Gibbs free energy function. And we can think of this function as being a function of the position and orientation of one protein, let's say the ligand with respect to the receptor. So the position is a 3-dimensional vector. And then the orientation is essentially, we have this body that can rotate around each one of the 3 axes. We have a rotation matrix, which is an element of, for those of you that are more mathematically oriented, of the so-called spatial orthogonal group. And we can parameterize this and we get a 3-dimensional vector. So there are 3 additional degrees of freedom. And because proteins are flexible molecules, so there is flexibility at the side chains, at the interface when they bind. There is also occasionally flexibility at the backbone level. We can include additional variables and potentially many such additional variables to account for this flexibility. From an optimization perspective, this is a hard optimization problem because we have a function that has many local minima. Some of these local minima are actually fairly deep, and we're looking for the global minimum. So typically what is done in the literature is what is called a multi-stage approach. So the idea is first you discretize these decision variables, if you will, and then you sample on this grid, and you sample with simplified energy functions using fast Fourier transform techniques. So in that way, you can score billions of conformations, and then you retain the low energy conformations and you cluster them. So you generate a number of putative clusters of protein conformations for a specific complex. And typically we can have as many as 30, 40, even more clusters for every specific complex. So given all of this, I will present three main ideas that we have contributed to this area. One first comment is that even locally minimizing such an energy function is not a simple exercise. We have developed manifold optimization techniques that make this optimization, this local optimization, more efficient than what would have otherwise been. Throughout, and as a side note, I will be referring to a number of papers, and where you see my initials, ICP, these are papers where my group has been involved. Now, moving beyond local minimization, we would like to achieve some sort of a global optimization objective. One idea is this idea of what we call semi-definite underestimation. So suppose that you have this function that has many local minima. You can essentially sample many different local minima of this function. So suppose you sample, and then you can generate a function which underestimates these local minima and minimizes the distance to that local minima. And we would like this function to be nice. So in optimization lingo, we would like this function to be convex. So you can think of this underestimator as essentially capturing the energy funnel of this binding energy for the proteins. So now, given the underestimator, then the SDU method, as we call it, alternates between additional sampling that improves the underestimator, and then biasing the sampling, we can easily compute the global minimum of the underestimator and then sample more in the area around the local minimum. The second idea is that of dimensionality reduction. So if for the time being we view these protein conformations as elements of R6, so rigid body translations and rotations of the receptor or the ligand with respect to the receptor, it turns out that if we sample low energy conformation, and we find that these conformations leave most of them in a permissive or a high variance subspace of R6. So essentially what is happening is that there are specific principal directions where if you plot low energy conformations along these principal directions, then you can see that they are very different from one another. But there are some other directions where if you plot low energy conformations along these other directions that live in the so-called restrictive subspace, then they would look very similar. So one is sort of falling on top of the other. And we have exploited this idea to refine this underestimation approach so that it takes place instead of R6 in the appropriate subspace of R6 that is defined by this permissive or high variance subspace. So to give you sort of an idea of, according to this analysis, how proteins interact and bind, you have a process of diffusion where the ligand finds the right pocket in the receptor, and then the rest of the binding takes place by essentially rolling this ligand on top of the receptor. And you can think of this as moving in this low dimensional, high variance subspace. So the third idea that we have contributed is this idea of ranking. So I mentioned earlier that these multi-stage methods produce many different clusters of possible conformations. Typically, one of these clusters will contain the true near native conformation, and it is of interest to identify among the multiple candidates what is this cluster that contains the native conformation. So at Boston University, we are hosting the most successful, most accurate automated protein docking server called ClassPro. So this has been led by Sandor Vazda, a good friend and a very close collaborator, and the development of ClassPro and the maintenance of ClassPro. And ClassPro implements this multi-stage approach, and then it ranks the clusters using the size of the cluster. So the motivation is to introduce in the ranking some surrogate for entropy, and this is more effective than ranking by energy. So in recent work that we just published, we developed a machine learning type of method for improving this ranking. So the idea is to use a robust regression to learn the quality difference between any two clusters. So if you give me the two clusters of the same complex, then using data, I can learn to rank order these two clusters. So now, given multiple clusters of a specific complex, through a sequence of pairwise comparisons, I can now rank these clusters and produce a final rank. And in results that we just published, we have shown that in most cases, the number of cases actually where the ranking, the near native conformation is ranked at the top of the rank increases by a factor of 2, by 100% compared to the ClassPro type of ranking that is based just on size. So I mentioned this notion of robust regression, and I would like to start, I guess, the first methodological digression. I'll take you through a little bit of a formulation and mathematics behind this idea, at least the way we think about this problem. So in general, in learning, particularly supervised learning, one is given a training set, and the training set consists of many examples and different examples where you are given a feature vector that characterizes every data point, and then you are given an outcome or a response variable. And what one is interested in doing is in learning a model. So this model is represented by this function. This model is parameterized by some parameter vector beta, so that for at least all data points that you provide in the training set, this function is close to the outcome, right? So we would like to predict the outcome. Now, many different setups can be incorporated in this formulation: classification, where the yi is discrete, or regression, where the yi is a real valued quantity. What is typically done, if we had information about the true distribution that generates the data points, so the learning then can essentially try and minimize this expected loss. So the loss function is a function that measures how well this function approximates the outcome. And if we knew the distribution, we could compute this expectation and minimize, with respect to the parameters of the model, the expected loss. Now, we don't know the distribution. However, we do have the training set. So the common idea is that we use the training set to infer a probability distribution, and specifically, in the absence of any other information, an empirical uniform distribution over the training set. So we end up minimizing this quantity here. This is this expectation with respect to the empirical uniform distribution over the training set. Now, these models, for instance, if they are linear, and this is the case that we'll mostly focus on, you have many common algorithms, ordinary least squares, least absolute deviation for regression, logistic regression for classification, that can be formulated in this specific way. We are interested in dealing with biological, medical data, and there are a number of challenges that arise in those domains. So one is the sparsity. So sometimes we may not have enough data. Sometimes even the variables that we have may be more than the data we have. So we are interested in this regime to avoid overfitting. Another issue is that many times, particularly in these types of data sets, the data that are given to us may contain outliers. So we need methods that are robust in the presence of these outliers. And finally, and importantly, one is interested in coming up with interpretable models. So because we want not only a prediction, but we also would like to understand why the model is making a specific prediction, so that if used by humans, by doctors, then they can develop some sort of a confidence in the predictive model. So I will talk a little bit about this notion of robustness and what may be a way of infusing robustness into the model. So here is the idea. So let me remind you that earlier we minimized this expected risk with respect to the uniform probability distribution over the training set. Suppose now that instead of taking the expectation with respect to that empirical distribution, I take the expectation with respect to some probability distribution Q. Let's say Q lives in some set omega. And then I compute the worst case loss, expected loss, over the collection of probability distributions Q, and then I select a model that minimizes that worst case loss. So you can think of this as a game theoretic setup of the learning problem, where you have essentially nature that selects this probability distribution Q, and nature is doing the worst possible thing for you. And we would like to have the last word and come and select a model that minimizes this expected loss, given the worst actions that nature can take. So this game theoretic, of course, approach goes back way back to the origins of game theory. And to put this in an optimization perspective, these are different flavors of optimization. So in a deterministic optimization setup, we have some function that we would like to optimize with respect to decision variables, and the data are given, are fixed, are deterministic. In a stochastic optimization setup, the data are not fixed, are generated from some known probability distribution, and we minimize this expectation. In a robust optimization setup, we allow the data not to be fixed, but to live in some ambiguity set. And then you see again this min-max formulation of the optimization problem that infuses this notion of robustness. And you can think of distributionally robust optimization as a combination of these two ideas. So data are generated by probability distributions, but we have an ambiguity set, an uncertainty set for these probability distributions, and then we solve a problem of this type. So I haven't talked about this set omega, so let me do that now. In the work that we have done, we consider this set to be a ball, a sphere of probability distributions that is centered around the empirical probability distribution that is induced from the training set. So that sphere is formed using some measure of distance between probability distributions. And that measure, that metric, is called in our work Wasserstein metric. So this has become very popular recently, and you can see a number of papers that have worked on this topic, including one of our papers. So let me give you a little bit more information behind that peculiar metric, the Wasserstein metric. So there are some other names that have been used: optimal mass transport, earth movers distance, and there is a reason for these names. So all of this goes back to Gaspard Monge, who was a mathematician and an engineer, was in fact later on a minister of the French Republic after the revolution, and he is considered to be the father of differential geometry. And he was thinking about a very practical problem. So if you have mines and you would like to send material from these mines to construction sites, how exactly you do that in order to minimize a transportation cost? And he formulated this problem. And then came Leonid Kantorovich, the great Leonid Kantorovich, who actually relaxed this problem and allowed for material from one mine to be split and potentially be sent to multiple construction sites. So now think of the mines as being one discrete probability distribution, and think of the construction sites as being a different discrete probability distribution. And you can formulate this transportation problem where this Sij is simply the transportation cost of transporting probability mass from location I to location J, and this Pij is simply the flow of probability mass along each one of these links. And these are simply mass conservation constraints. So this is a linear programming problem. It's a transportation problem, very well known in the operations research literature. And the optimal value of this problem is the order 1 Wasserstein distance between these two discrete probability distributions. So all of this, of course, can be extended in great generality, can be done in the continuous case. This is the metric that we are using in formulating this robust problem. And what we have shown is that if you now have your empirical probability measure over the training set, and suppose that you introduce some outliers that are generated from a probability distribution that is different from the true probability distribution that generates most of the points, we have shown that you can find a radius of that Wasserstein ball so that the true probability distribution is included where the outlying probability distribution is excluded. So that gives you a sense of robustness of this approach. And we have also shown that the problem is not much harder than the original empirical risk minimization problem. You can relax this problem, and instead of solving the min max through duality, you solve a problem that includes the standard empirical risk and then includes a regularizer. And this regularizer is expressed in terms of the dual norm of the norm that you use to define your Wasserstein metric, so this transportation cost. So this is, I think, interesting because it connects regularization with robustness. There are strong out-of-sample generalization guarantees that one can show. This norm can be very general, and also there are extensions to multi-output regression and multi-class classification. And the past year, one of my former students, Ruidi Chen, and I, we wrote a monograph where we put all of our work in that space and work of others, including in this monograph. Okay, so we are roughly here at our first stop, and then we are on our way to the second stop in this adventure tour. So I will talk about bacterial cells. So this is a metabolic network that corresponds to E. coli. E. coli is a bacterium. And within the cell, there are many chemical reactions that take place. And these are the chemical reactions that produce all molecules that are needed to sustain life. So for the cell to multiply and for the culture or the community of cells, bacterial cells to grow and the community to thrive. So we can think of the cell as a chemical factory. And the cell has to make decisions about what are the items and the quantities of these items that need to be produced. And it is of interest to develop some model in which we can predict metabolic activity, and that may be useful in understanding how biology works at this level, but it may also be interesting in case we would like to change this chemical factory for our own purposes. And today we have that ability. So to give you a very contemporary example, the mRNA COVID vaccines are produced using bacterial factories. So the Pfizer vaccine, DNA of the spike protein gets inserted in E. coli, and then the E. coli multiplies, so you have mass production of the DNA of the spike protein, and then this is filtered, and then gets translated into mRNA, which is what gets in the solution that goes into your arm. So in one of, I would say, remarkable theories that have been developed in the last 15 years or so due to Bernhard Paulson, he postulated that the cell is actually solving a production planning problem in order to make decisions about what to produce and at what quantities. So here is this formulation. X here are the fluxes of the chemical reactions that take place within the cell. This S is what is known as the stoichiometry metrics. So these are essentially mass balance constraints in steady state. There are some constraints that are affected because of the growth medium where the cell grows. And then you have this peculiar objective function that somehow captures fitness, right? So captures the ability of the cell to survive and multiply, which in the field is called biomass. And there has been, I think, a long-standing question in the community: what is this right objective function? Is this constant? Does it changes over time, depending on the environment where the cell is, and so on? So we have developed an approach to be able to answer this question. And this approach essentially tries to reverse engineer what is happening within the cell. So suppose that I give you experimental data. Suppose that you can measure what's going on. You can measure these chemical reaction fluxes. And you have also an idea of the chemical reactions that take place, or the structure of the metabolic network. You also know the growth medium where the cell is growing. Can you infer, if I give you lots of data, lots of measurements, can you infer what is the cost function that drives this process? So we have, if you can think of this problem as a very simple instance of a more general inverse equilibrium problem, and together with Dimitris Bertsimas and Vishal Gupta, who have developed a theory of how you can solve these inverse equilibrium problems. And I will not get into many mathematical details, but essentially for those of you interested, this can be formulated, the original equilibrium problem, as a variational inequality problem. And we have developed a way, given data, given solutions to this variational inequality problem, to recover the common cost function, and we assume it is common, that drives all of the solutions, that explains all of the solutions. And together with Daniel Segre and his lab, another very close collaborator here at BU, we have applied this machinery and adapted to answer this question in metabolic networks. And here are some experimental results. So in this graph, and it's not so interesting what this graph is, but you can plot here an ancestral E. coli strain. And these are all strains that have evolved from this ancestral strain. And you can see that, at least in this graph, there is a clear separation between these strains and those strains that somehow have become a bit more efficient in terms of growing. And there was a speculation in the community that, this divergence may potentially imply that there is a divergence for optimality from this principle of optimality that I mentioned earlier. And what our argument has been, and what we've been able to show, is that this difference, this separation, can be explained by differences in the objective functions that these types of strains are used, that are different from the objective function that these blue strains are using. So it's not that we move away from optimality, it is that the cells have a more complex objective function in terms of solving that production planning problem. So we are roughly here, and then for the second half of the talk, roughly, I will talk about humans, more complex organisms, and some applications in medicine. So in my group, we have done quite a bit of work working on chronic diseases. So specifically, we focused on heart diseases, diabetes, and also surgical outcomes. And we have developed a variety of models that predict important events. So these events are in the works that we have done. hospitalizations, readmissions, prescribing treatment. So these are prescriptive models that decide what is the right treatment in order to optimize future outcomes. We'll not talk about this work because, as you probably know, this past year, the entire world has been preoccupied with one thing, and these pictures pretty much say what that is, COVID. So we have shifted gear during this past year, and we have worked with a number of different COVID data sets, working with a number of different groups across the globe, including a group out of Mass General led by Dr. Velmachos, using patients from Mass General and Brigham and several other Massachusetts area hospitals. We have worked with a group in China, working with data sets that came out of the epicenter, the original epicenter of the epidemic in Wuhan. And we've also worked on nationwide data sets. So a data set from Mexico with my student Salo Wallenstein and Christos Cassandras. And then another data set out of Brazil, again with Salo and Christos, and one of our former students who is in Brazil. So I will not talk about these either. I will talk about a bit more recent work that we are now completing. So we haven't published this yet. And this is working with collaborators at the Boston Medical Center at the BU School of Medicine. So we recently got access to the entire data set of patients who tested positive for COVID-19 during 2020 at BMC, 7,000 roughly patients throughout 2020. To give you a sense of the basic statistics, what happens to these patients, about 19.5% of those get admitted to the hospital, to BMC. Of those admitted, 24% end up in the ICU. Of those in the ICU, 58.7% get intubated. They are hooked up to a mechanical ventilator. And unfortunately, 70% roughly of those intubated actually pass away from COVID. So for these patients, we have lots of information. We have demographics, we have vitals, radiology reports, medical history. We have more than 2,000 different labs. We have information on more than 3,800 medications that are given either prior or during the hospitalization. So there is quite a bit of data engineering that is required in order to get to the point of starting to develop models. What is interesting about this data set is that we have information about social determinants of health. So BMC patients are screened for their need for housing, need for food, for transportation, education, ability to pay for utilities, and other social needs. So we have that information. And we also have information on how busy was the hospital at the time that each one of these patients was diagnosed positive with COVID-19. There is quite a bit of natural language processing that we have to do. This type of data are not really easy to deal with. They are not in a tabular format. So particularly in this case, radiology reports are given to us in the form of text. So we have to extract using NLP appropriate features that can then be used to feed our models. And what we did is we developed models that predict hospitalization, predict whether someone will need ICU treatment, mechanical ventilation, and eventually death. And I will not, in the interest of time, walk you through all of these models, just only mention one model, the hospitalization model. So short story, this model can be very accurate, even with relatively few variables. We get AUCs, so area under the curve, of about 92%. These are out of sample on a test set. It is interesting to take a look at the variables that are driving this decision. So these are the most important variables that determine whether someone will be hospitalized or not. So you see some variables that correspond to pre-existing conditions that in most cases increase the likelihood of a hospitalization, in some cases may slightly decrease the likelihood of a hospitalization. It is also interesting that the social determinants of health come up. So need for food and need for transportation are important variables and they contribute to a hospitalization, potentially because of malnutrition or because of the quality of the diet, maybe because of lack to your own transportation and the need to take public transportation and the type of viral loads that you may be exposed, and because these may also indicate other issues with the patients. The role of social determinants of health also came up in our study of the nationwide Brazilian data set. So there we found that there are social variables that also impact significantly the likelihood of severe disease. What is also interesting is that the occupancy of the hospital is a factor that decreases the chance of hospitalization. And it makes sense if the hospital is very busy, potentially more stringent criteria are being used to decide to admit a patient in the hospital versus not. What we found that is also interesting in this case is that in the BMC data set, and these are in the test set, so these are test set numbers that I'm showing, we have roughly the same number of blacks as number of whites. And notice that the false positive rate for blacks is twice as much as the false positive rate for whites. And correspondingly, the false negative rate for blacks is lower than the false negative rate for whites. So you can think that essentially the model learns to be biased. And if someone is black, the model is much more eager to predict that this person will be hospitalized because of COVID. And I guess that's another indication that being black is not only simply a matter of the color of your skin, but is more of a social construct that comes with additional variables that potentially are in the form of pre-existing conditions, social determinants of health, or even other variables that we don't observe that somehow indicate that the likelihood or the chance of being admitted to the hospital is significantly higher if you are black, and the model becomes biased in this way so that it improves the accuracy of the model. So this is the mortality model, and in the interest of time, I'm going to skip it other than say that is extremely accurate and much better than other alternatives. We have developed calculators that we've made available in our lab's website for people to use in the community. And I guess a take-home message is that COVID is a very different clinical signature than other forms of similar diseases, of other pneumonias. And in some instances, the models can actually surprise even top doctors like our friends at MGH. So for instance, here is a case of a patient, 51-year-old, I believe at MGH, that showed up at the hospital, was hospitalized, but was doing quite well. So had high oxygen concentration, was doing well for 3 days, and then all of a sudden gets admitted to the ICU, gets ventilated for 30 days, and the model was able to predict the ICU admission upon admission of the patient to the hospital, simply because one of the labs, this is lactate dehydrogenase, was high, and the patient was a diabetes patient. Okay. I made a comment earlier about the heavy use of natural language processing in health applications. In our work with some of my former and current students, Henkhui Zhu, Boran Hao, Amir Tamashebi, who is a collaborator, we have developed ways of infusing biomedical knowledge into generic language models. So language models that you can train from generic text, let's say Wikipedia. We have developed some methods that we can make these models a bit more biomedically aware. And in some recent and to me very exciting work, we are working with people at the Framingham Heart Study, Rhoda Au and her group, and one of my current students, Amada Mini. We are developing a way of automating Alzheimer's and dementia review. So subjects, patients in the Framingham Heart Study, if they show signs of cognitive decline, then they are subjected to a very rigorous and very lengthy dementia review. So this is a neuropsychological review. They essentially take them through a series of cognitive tests, about 8 or so tests, and this interview gets recorded. So we have digital voice recordings of this interview. And then after this interview, a very specialized clinician reviews the entire record for the subject and makes a determination whether the subject is normal or suffers from mild cognitive decline or has dementia, and there are different grades of dementia. So we want to automate this process. And our solution is we take these voice recordings, we transcribe them from voice to text, and you can use very standard platforms. Google can do that for you. And then that's where from text, NLP takes over. So we diarize this document, the transcription. So separate who is the subject, who is the person that is conducting the interview. We also using NLP to separate the different sections of the interview depending on the test that the subject undergoes at that time. And then we develop sentence encoders that come out of the sentences. And we throw all of this into a machine learning model, different types of models, to train the model and be able to recognize normal versus dementia. And these are some preliminary results, so these are not yet published. So if you just try to make this prediction simply based on demographic information, so age, race, sex, and so on, you get an accuracy AUC that is on the order of 74, 75%. If you throw in genetic information that again may not be available for everybody, there are specific genes that are associated with Alzheimer's, then that improves a bit, you get maybe to 77%. If you use this approach and combine it with the demographic information that is easily available for everybody, you get AUCs that are on the order of 90 to 93%. So almost as well as this very elaborate review that a specialized clinician would do. And that is potentially exciting because of course, access to this level of attention and treatment is not available for everybody, even in the US, and is definitely not available for most people in other less developed countries. So that may be an avenue for making this more globally available. So in the final part of this presentation, I will just talk a little bit about learning dynamic policies from data. There are many reasons for doing this. So far we were considering problems that you can think of them as static, right? So there is information that I have. Based on this information, I would like to make a prediction, and that's it. In a dynamic setting, you make a decision, and then the decision alters the state of the system. And then you have to make another decision, which is going to further alter the state of the system. So you are interacting with the system. And this could be of interest in a healthcare setting. So for instance, most treatments are dynamic, right? You make a decision, could be a medication or some other type of decision, and then the state of the disease evolves, and then you have to continue be adjusting your decisions so that you impact future outcomes. Developing such dynamic policies from scratch, particularly if you don't have a detailed model of how the system works, is very hard. So this is what Bellman called the curse of dimensionality. So we talked about the static setting. We have developed some ways of using data to learn policies from that data. So again, in a healthcare setting, you may observe good doctors at MGH, at BMC, at some of our best hospitals. And you can try and learn what are the policies simply by observing the doctors. This has also other implications in automation. Suppose that you want to guide a UAV in a complex terrain. Again, you would like to learn a navigation policy. And there are animals, there are humans that are expert navigators, so there is an opportunity to learn from them. So there are, I would say, two main approaches that have been used in the literature. One is called inverse reinforcement learning, that essentially learns a reward function from your observations. But the drawback is that you don't avoid having to solve that hard dynamic optimization problem for finding an optimal policy. You don't avoid the curse of dimensionality by doing this. Another approach is what is called imitation learning. So what if we make these observations and then we can learn a policy directly from these observations? So we have done work in this space, learning policies for Markov decision processes directly in papers that we have published recently. And more recently, we have developed methods that learn how to develop hierarchical policies with my student Ziyu Zhang and others. And there are many good reasons why you may want to impose some sort of a hierarchy. After all, human beings, animals, they have, at least human beings, they have a hierarchical way of thinking, and imposing that structure may be beneficial in several domains. So what I would like to focus a bit more is, suppose you learn such a policy. How can you improve the policy? And essentially the question is, how can you learn from your own data? And just to formulate a bit the problem, suppose that we have a Markov decision process, and we have some parametric policy. So this policy is parameterized by a parameter vector theta that maps states of the system to actions or other probability distributions of their actions more generally. And the way in which so-called policy optimization approaches work, at least using data, is the following. Suppose you now have this current policy, you play this policy, right? So you exploit the policy and you collect trajectories that essentially tell you how well the policy is doing. So if you could now find a way from this information to improve the policy, then that's a good step. And you keep taking these good steps until you cannot improve the policy much further. And your overall objective is to maximize this long-term discounted, in this case, reward function. So I will briefly describe a method that we have developed. We call it uncertainty-aware trust region policy optimization. This is a paper that we recently published in AAAI with my student Jimmy Queenie and Christos Kassandras. We co-advise him. And the idea is the following, comes from a so-called policy improvement lower bound. It goes back to ideas on so-called conservative policy iteration due to Kakadi and Langford, and more recently in this trust region policy optimization by several people that was proposed. So this idea is the following. Suppose that theta k is our current policy, the parameters that parameterize our current policy. Now, the performance of any other policy is given by this quantity. So we would like to find a new theta that maximizes this quantity. But because it's hard to maximize this, instead we will try and maximize an appropriate lower bound. So in this lower bound, this G, you can think of this as being a gradient of your performance metric with respect to the parameter vector. And this F, and it's not so important, it's called the Fisher information matrix. So the TRPO, trust region policy optimization approach, essentially maximizes the lower bound by maximizing this quantity subject to some quadratic constraint that defines an ellipsoid, defines a trust region. And the idea is not to move too far from your current policy, because all of these are estimates based on data, and you have uncertainty, right? So you don't want to take a direction that will take you down the wrong alley, down the wrong path. So our idea is to try and robustify this optimization. So rather than optimizing this quantity subject to these constraints, suppose that we say that this gradient vector lives in some uncertainty set. So now we will compute the worst case version of this bound, and instead optimize this quantity. And that relates back to the robust optimization idea that and approaches that I mentioned earlier. So it turns out that you could construct uncertainty sets that make sense. And also it turns out that you can now solve this problem, this max mean problem. And what you get is a policy improvement step that is very similar to TRPO, but is uncertainty aware. So these is an estimate of the gradient vector. This is now a new matrix that is a bit different than the estimate of these metrics here. So again, you maximize the linear function, and you have a quadratic constraint. So that's a modified trust region. And these metrics includes the estimate of the Fisher information matrix plus something that is proportional to the covariance matrix of the gradient vector. So now this is aware of the fact that you may have limited data, that you have lots of uncertainty, which is reflected in this covariance matrix, the level of uncertainty, and also in which directions you may have more certainty, in which directions you may have more uncertainty. I will not obviously take you through mathematical derivations other than this at least a key idea behind the approach, but I will show you some experimental results, some simulations. So these are these locomotion tasks, and the underlying policy is represented using a neural network, and the details are not so important. So I will show you this guy. So this is a walker, and the objective is just to walk fast and walk as fast as you can over this flat terrain. And you will see here the walker using TRPO, and then the walker using this UA TRPO, which is our method. And this is in the case where you have lots of data, there's not a lot of uncertainty, there's not a lot of noise in the environment, and so on. So you can see that both fellows are actually able to walk very fast and go far, and essentially both policies perform similarly in this setup. Okay. So now let's make things a little bit harder. Suppose that either I don't have lots of long trajectories based on which I compute my policy updates, or suppose that there are other issues. Maybe the ground is shifting underneath you, or maybe there are sensory motor issues. You don't feel your legs very well, or you cannot process because of some disability the information that comes to your brain in making, guiding your legs. So in this setup, the question is how does TRPO does and how this UA TRPO does. And you will see that obviously they walk now a bit differently, right? So that's probably how you would walk if you felt that the ground is shifting underneath you or it's very slippery. But this fellow is able to walk, but not very well. You will see that it falls, but this fellow manages to walk and without falling. So this is the benefit of being uncertainty aware and robust. And over many runs, the average performance of this uncertainty aware policy is significantly better than the more standard earlier policy. Okay, so that's what I had for you today. You will allow me a few minutes of acknowledgements. So first of all, thanking people that pay the bills. I would like to thank many collaborators on the protein docking work, on the inverse optimization and metabolic networks work, many colleagues at Boston Medical Center, the School of Public Health at BU and Framingham are mentioned here that we have collaborated. And we continue to collaborate, colleagues at Mass General that have been good friends and collaborators, people in China that we collaborate in this COVID project, Julia Fleck in Brazil. And I'm also listing some of my close BU collaborators. I have not been able to fit our joint work in this presentation, but in my defense, I was given only 1 hour. I would also like to thank my students. So they say it takes a village, and this is my village. So this is my lab. And these are students whose work was mentioned during the presentation. Their papers were mentioned. Alumni of the lab and also current students. I'm very grateful for all of their contributions and all of their work. I would also like to thank all of the alumni of my lab. I have learned a lot from them, and I really enjoyed working with each one of them. It takes mentors, so I'm grateful to two individuals, Dimitris Bertsimas and John Tsitsiklis at MIT, that were my advisors, and I consider them people that have learned so much from them, and I continue to learn. And they have been great role models and very good friends. And also I would like to thank three individuals at BU that have acted as senior mentors, great friends and collaborators, specifically John Belial, Michael Karamanis, and Christos Kassandras. Thank you to all. And finally, it takes a family. So this is my family, my amazing wife, Gina, and our sons from left to right, Aris, Phivos, and Alexandros. I wouldn't have been able to do anything without them. Thank you so much.
Thank you, Giannis. A virtual hand for a fantastic talk. You've got everyone on this can go ahead and enter either in the chat or in the Q&A questions for Giannis.
I want to correct that and say put it in the Q&A, not the chat.
Yeah, I was not able to put my one question in the Q&A because I'm a panelist, not a attendee.
Oh, you can only answer things. Okay, so let's kick it off. David Boas has a question. He says, the work with Rhonda Au is tremendously exciting and with high impact for patient management and future drug studies, for instance. What are your next steps with that project?
Yes, thank you, David, for this question. It's a wonderful question and is exactly what we have been thinking. The objective is to make these types of reviews much more broadly available and also be able to make them remote, so not have to bring patients into the clinic. So we are working essentially with so-called digital information, either in the form of voice, or we have also information from people drawing simple images. And it's amazing that just based on these images, you can determine whether someone has dementia or is normal. And part of the objective is if this becomes much more broadly available, then you have a much larger community of potential targets for clinical trials, for Alzheimer's, because as you probably know, the vast majority, maybe 100% of all Alzheimer's trials fail. And one of the potential reasons may be that they are targeting the wrong individuals. So having broader testing and review would be very helpful.
Okay, thank you for that. Diane Joseph McCarthy asks, very interesting model predicting hospitalization with COVID. Is it correct that the model indicates COPD decreases the likelihood of hospitalization? And is that surprising?
That's a great observation, great catch. Indeed, that's the case, at least in the BMC data set. And it is a bit surprising. There have been in the literature some other papers that come to the same conclusion. And I don't know whether the mechanism is well known, but potentially a plausible explanation may be that there are quite a lot of damaged tissue in your lungs if you have COPD, and then that doesn't give enough ground for COVID to invade, for the virus to invade. That may be an explanation.
Great, thank you. John put a question in. It says, brilliant. So I'm not sure what that question means, but you can answer it any way you like.
Thank you, John.
Okay, Ken had put a question into the chat, so I'll pass that one up. He wanted to know, what does it mean for a predictive variable to be negative or positive? For example, SpO2 was -0.8 and transplant was +0.7.
Yeah, so if the sign is positive, it means that elevated values of that variable increase the chance for hospitalization, and if it's negative, they decrease the likelihood of a hospitalization. So you would expect that the higher the oxygen concentration, the better you are and the less likely you are to be hospitalized. Whether if you are a transplant patient, then that will also increase your chance of hospitalization.
Okay, Ken, can I ask your second question?
Go ahead, and then we don't do the answer. I'll just give you a few. But yeah, go ahead. Pick the one you like.
Okay. For your cell metabolism results, what does it mean that a set of cells is now using a new objective function for the same glucose conditions?
Can you repeat that? I missed.
For the cell metabolism results, what does it mean that a set of cells is using a new objective function for the same conditions? You were talking about this scatter.
It's not clear what led to this different objective function. It may be conditions in the slightly different conditions in the environment during the time that the experiment was done, and you had evolution to that new strain, and it may be other random factors. But I guess the point is that now, based on experiments, one can understand a bit more, get a better handle of what is the objective function that is driving the cell. And also that objective function, we have seen in other results with Daniel Segre, that it may change over time as, for instance, the conditions change over time.
Interesting.
Okay, question from Chris Chen. Amazing work. I'm curious, given your work across so many different fields, what you feel is rate limiting in your work. Developing models, gathering, organizing data so you can plug it into the models, getting more experimental data, math, ...
Data, money, and students.
You seem to have plenty of students and money, so maybe it's data. Okay, question from Sandor. In 2021, it was shown by DeepMind that machine learning provides higher accuracy for protein structure prediction than the traditional optimization of the free energy. How will this change your research directions?
So Sandor, I think you know very well the answer to this question, and we are jointly working on these fronts. So I think there is potential for using more sophisticated end-to-end machine learning problems, approaches, potentially using natural language processing in improving these predictions that are solely for the time being based on energy. What DeepMind has done has been demonstrated for folding, but I think there are good prospects for potentially doing work in the docking space as well.
Okay, we have no open questions.
Okay. So turning it back to me, we're done with the questions, right?
Yep.
Okay. Well, I want to thank Giannis very much. I wish we could have done this live. Wish we could have had the post-talk reception and shaken your hand and had a glass of wine, some hors d'oeuvres, and we will some way get back to that. We do have a gift for you. It's a captain's chair with engravings celebrating this wonderful. Oh, you have it already. There it is.
I have it. Yes.
There it is. I was going to tell Teresa to take a photograph of it before we sent it over to you, but she was too good.
There you go. Thank you so much.
Plus a framed version of the talk, which we'll also put outside the dean's office.
Absolutely. Thank you for the poster and the great chair. Appreciate it.
Well, the college thanks you for your extraordinary research success and impact, Giannis, and for your collegiality and your willingness to do this in a virtual way. And this was really, I'm fascinated by a bunch of stuff I'll talk to you offline about. Everyone, thank you for joining. Thank you for participating. And we will move on to next year's. And Charles, thank you if you're listening. Everyone have a nice night, and we'll see you, many of you, next week at the faculty meeting at the graduation and commencement receptions. Thank you, and take care, everyone.